Hi everyone, my name is Victor, and today I will present some approach that we think in this approach last week. And this approach is start with a research of growing epithelium in these images that you can see in the first layer. It's a start at time 0. Simulation of real cells living and growing. When some cells grow, they need to multiply and divide. So in this experiment, they use some method that I showed to you in the previous seminar when I speak about machine scientists. The method created by Roger Guimera, and in this article called Regulation of Cell Cycle Progression by Cell and Cell Matrix Forces, Roger Guimera gave some contribution. So I will say, in my presentation, I will say to you the Roger Guimera contribution for this work, and then my recently created approach. Here we can see 3 layers. In the first layer is just the cells in a fluorescent, with a fluorescent light. In the second B layers, we have the same, but here we have these lights represent the traction, and here represents the tension. I will say, what is this? Here we have a zoom in this group of cells, and here we have the neighbor cells of one cell that was being divided. And so this cell was passing through a mitosis. And then we have here some the surface area about the divisions, their division over time. And the division takes 33 minutes to end and form 2 cells. In the next image, I show only the cells. And when we, and here we can see the one cell turn to be 2 cells. And this is the particular change in the area of the cells. They used the area to understand these changes and mitosis. And using the area of neighbors, we can measure the traction, what they call the traction between the neighbors and the cell that is being divided. So here they can use MATLAB. They use MATLAB to measure some pressures between the neighbors and the cell. And here we have an example of this. So machine scientists start by Roger Guimera contributes to this work, finding these two functions here. Using all the layer of cells, they use machine scientists to obtain this function of traction, that is the pressure inside the cell, and the tension, that's a function that fits the pressure of the cells and the neighbors. So in this approach, they use images and areas to do that. And this is a different approach that I was looking for. So I read in this little comment here in the Nature, this article only says that in their methods, they only say this shortly text to linear model selection analysis, and then they just say, oh, we use machine scientists to do this, and this is the results. So my approach, in our recently created the approach, we have this signal. This is from a cell that passed through the division, but it's not a lot of cells, it's just one cell. And this cell emits some intensity of fluorescent light, and this is their graphs. So their graph. Here we have a conjectural question, because we think that when the cell starts to divide, they change our intensity of fluorescent light. And here you can see the intensity decrease. And we should find a method to say where the cell starts to prepare for division, like a prelude of division, not when they are dividing, because if the cell is being divided, we can see the area just growing bigger. But here we can say, oh, it will start to divide. It's when this cell will start to prepare itself. So our approach is using this data, we simulate another data just to see if our approach will work. We get here a scene and a dumped scene, just like to see, and we get two functions about, use that function, we separate two functions, and each of them we will represent like this, yes, and then use these two functions to train a neural network. Neural network will fit all the points of the scene and all the points of dumped scene, and then we will save this error value. So that's okay, that's nothing. But if we do another separation, like here, for example, we have two different functions, and then the first function starts to be a scene, and then be turned into a dumped scene, and the second function is the same. But the question is, our approaches, this is the main idea of our approaches. When we do different separation of two functions, we have the mean error bigger than before. So our approach is we will forget where we made the two functions, and we will randomly choose a separation and then train two neural networks, and then we will compare with the best model. And when we have the mean error, we think that will be in this division that form two simple functions. And that's our approach. Unfortunately, in conclusion, we are hopeful to this new approach, excited to work on contributions to better understand cell divisions. And we start to work on this approach a few days ago. And if you want to see the future results, you can follow the GitHub page. And that's it.